Robbie comes to us after doing a lot of traveling throughout his education. He started out by getting a BS in biology and a BA in international studies at Oregon State University, and then a master's of science in tropical ecology from Universidad San Francisco de Quito. And then a PhD in ecology and evolutionary biology from Brown University, followed up by postdoctoral appointments both at Woods Hole Oceanographic Institution and Brown University. And Robbie has really diverse interests. A lot of the main ones I listed here, from what I could tell from his publication record and from what I know about Robbie, is reef fish ecology, coral reefs, kelp forests, climate change ecology, marine diseases, and endangered and endemic species. Of course, that's not an exhaustive list, but just some of the highlights. And of course, you see Robbie here holding a snook, which you'll hear a little bit more about. So, without any further ado, I will yield the floor to you, Robbie. Thank you for being here.
Thank you, Savannah. Thank you for that wonderful introduction. Thank you both to Laura and Fia as well for managing this series. It's something that I attend religiously every month, and I really look forward to it. It's a great exposure to the people, the nature, and the science that's going on along the Nature Coast. So thank you again for this great invitation. So today I'm going to be talking to you guys about some of the work that I've been doing in my lab and several of my students have been contributing to, which is trying to understand the importance of nursery habitats for juvenile sport fishes in the Nature Coast. And one of the reasons that I think that this is a really important thing to study and perhaps something that a lot of people haven't done yet is the fact that, well, first off, these nursery habitats for juvenile sport fishes are very hard to access. They tend to be in small tidal creeks and ponds, way up in the estuary that are very hard to get to. But also, our focus is usually on these big sport fishes, which you can see my student Max holding a nice 40-inch snook there off the coast of Cedar Key. But the reality is that if we want to have access to healthy and productive fisheries and all of the economic benefits that come along with that, we really need to know what is going on with the juveniles and those larval fishes, the really small ones, where they're growing up, what kind of conditions they require to live and grow, and how they integrate into the adult population. That sort of pairwise reciprocal gene flow between spawning adults and juveniles growing up in nursery habitats. So this work is funded in part by Florida Sea Grant and the NOAA Restore Science program. We also receive funding from the National Science Foundation, and this is primarily a partnership between the Nature Coast Biological Station in Cedar Key and the School of Forest Fisheries and Geomatic Sciences at University of Florida. And we also do a lot of work with Florida Fish and Wildlife Conservation Commission, and that will become evident here. So, where do juvenile sport fishes grow up? Again, if we want to be able to catch those big fish eventually, we need to know where those juveniles are growing up. And a lot of sport fishes along the Florida Gulf Coast have a bipartite life history. Bipartite meaning they have two different age classes that exist in spatially different areas. So this is an example from Sulor Barberi and et al.'s 2020 study on gag grouper. But a lot of species that we'll be discussing today have this sort of bipartite life history, where you have spawning adults that are somewhere offshore. Gag grouper are going to be in the greater than 50-meter depth range as adults, but there are other species like snook that have more inshore adult life history stages. But this sort of offshore adult life history stage is when they're sort of at their peak size. This is when they're spawning. They get together oftentimes and spawn in spawning aggregations. Um, Gametes are fertilized oftentimes in the water column, and those gametes then develop into eggs and larvae. Those larvae eventually hatch out, and after several weeks to months, they settle down into nursery habitats along the coast. And those nursery habitats are often seagrasses, salt marsh, mangroves, oyster reefs, but generally they are shallow and very, very far inshore. And the reason for this is that it's really hard to survive in the open ocean or in deep reef habitats. There's just a lot of predators. And so by having their larvae and juveniles grow up in these inshore coastal habitats, fish can get away from some of that predation pressure in those really, really small size classes. But that also means that those small size classes need access to quality nursery habitats. And in the context of what we're experiencing in Florida now, the phenomenon of tropicalization, this is becoming a much more poignant issue because those nursery habitats themselves are changing. So tropicalization is a phenomenon arising from modern climate change, and it is characterized by range expansions of tropical and subtropical species into temperate species normal ranges. And that's coinciding with the contraction of many temperate species. And we see this both in foundation species such as mangroves and also the species that follow those habitats, such as sportfishes. So this results from the increase in average summer conditions, so average temperatures, but also in the increase in the frequency and intensity of extreme heat events, such as marine heat waves. At the same time as in the wintertime, we have also an increase in average temperature and a relative decrease in the frequency and intensity of cold snaps. So there are some species that cannot withstand those really, really cold temperatures. There are some species that cannot withstand really, really warm temperatures. And somewhere in between is how species distributions are dictated. And this isn't necessarily just the response to environmental conditions, but it also interacts with species interactions such as competition. So it might not be lethal for a species, but it might reduce their competitive edge that they have against other species. So two of the sort of poster children of tropicalization in the Florida Gulf Coast are mangroves and snook. Mangroves have been expanding since at least the 1950s. Snook more recently has been described as expanding north of its historic range in Tarpon Springs. And today we can see snook all the way up into Apalachicola. So the combination of sportfish range expansions and changes in the distribution of the habitats that make up nursery habitats where juveniles of these sportfishes grow up has called this research into being, has sort of necessitated this research as both phenomena are happening simultaneously. And I think it's relevant to bring in here the fact that these are not just interesting species for scientists, nor are they simply something that people want to fish. These are actually changing fundamental processes and economics of these regions. For instance, in Cedar Key, a lot of fishing charter guides are actually actively changing their methods to target snook, which is a resource that did not exist in Cedar Key 20 years ago. And you can think of many other examples for mangroves. So ultimately, these are really important changes, not just from a scientific perspective, but also for human economics and other uses. So why do we care? Well, fisheries, we want our fisheries to be productive, not just today, but long into the future. And the management of those fisheries and sustaining their productivity necessitates an understanding of nursery habitats for the juveniles of those species. Where do they exist? What are the conditions that facilitate growth and survival of juveniles? And also, what is the degree of population connectivity? Just because you observe a nursery habitat in a particular location doesn't necessarily mean that those are the offspring of nearby spawning adults, because currents bring larvae and other propagules all around the Gulf. And there might be bottlenecks, places where they don't survive well or where they are unable to migrate out into those adult offshore populations. Another big issue that is really important for the Nature Coast in particular is the fact that if we can identify these important nursery habitats, those could actually become permanent conservation lands in the future. This is a unique opportunity in the Nature Coast because it is a fairly undeveloped section of Florida's coastline. And as I mentioned before, snook are actually becoming a part of Cedar Key's identity, and mangroves have been for many, many decades. So these changes become integrated into local communities in fundamental ways. And so again, This is why it's really important to identify these nursery habitats and track them over time. And again, in the context of these range expansions and tropicalization, our main questions are as these estuarine foundation species and the sport fishes themselves are changing in their distribution due to tropicalization. One, how do juvenile sport fishes overlap in spatial distribution and diet? This is kind of touching on that question of competition. If juvenile snook are suddenly existing in a place where they did not exist previously, they may be competing with other native sport fishes to that habitat in utilizing those nursery habitats and having access to food and refuges from predators and so on. Secondly, which coastal habitats and environmental conditions are supporting juvenile sportfish nurseries and for which species? This allows us to define what the sort of parameters are for a healthy nursery habitat for each species and gives us targets for either restoration or long-term maintenance of those conditions in order to support healthy populations of fish. And then finally, do these juvenile sport fishes vary in their spatial distributions according to season and their tropical affinity? So if these species are tropical versus temperate. And this gets into questions of what are the microhabitats available in nurseries and how are they being utilized by different species based on their tropicalization status. So as I mentioned, a lot of these locations are hard to access. They're way up tidal creeks that are very shallow, have a lot of oyster bars and bulkheads. And so oftentimes we would have to canoe. And I think our longest canoe was something like 20 kilometers in one day. So just getting to these places is really challenging. But it's also really rewarding. We got to see some really pristine salt marshes and places that people just don't go very often. We use a variety of methods to collect our information. We did a lot of seining. We used a 9-meter center bag seine that was deployed along shore. So one person would stand with that one end of the net on a PVC pipe. The other person would walk out a couple of feet and then along shore and then back in. And those two ends of the net would be cinched together. And that way we could estimate density of fish within that prescribed area. We identified every fish to species, did counts and sizes of each individual, and we also kept fin clips for snook in particular because snook were trying to do a genetic survey to examine this pairwise reciprocity. Again, this degree to which locally spawning adults around Cedar Key are contributing to the juveniles that are growing up in Cedar Key, and which of those juveniles growing up in Cedar Key are recruiting into that adult population, growing up and migrating offshore and joining those adult spawning aggregations. You would assume that that's the case, but nobody's actually tested it before. And since this is a novel range expanding population, it's relatively small in size, we believe. And that makes it somewhat easier to do these pairwise genetic analyses where we're trying to match adults with their offspring. And we also took water quality parameters and habitat metrics to try to understand what are the conditions that really facilitate a healthy nursery habitat. So for the environmental data, we collected both in situ long-term data on temperature. So that's using an onset hobo pendant temperature logger. We zip tied that into a PVC stake and staked it down into the marsh at low tide so that we could get a long-term measure of how temperatures vary over the course of the year. And then we also used in situ YSI sondes, and that gave us instantaneous temperature in addition to dissolved oxygen, conductivity, salinity. These are various metrics to describe sort of water quality parameters and water chemistry. And again, fish communities were surveyed using these seines. We did this all throughout last year, starting around July and into February of 2026. And now I want to bring up this concept of thermal refugia. And this is really important for a species like snook because thermal refugia are microhabitats that maintain different temperatures than ambient conditions. There are many different types of thermal refugia in the world. In the ocean, deep ocean or deep reefs are a thermal refuge because shallow water tends to warm up faster than deeper water. And in North Florida, uniquely to North Florida, we have a large number of springs. So springs, as we all know, are areas where the Floridian aquifer is discharging up to the surface. They create many large springs that emanate into their own rivers, such as Manatee Springs that you see in the top right there. But they also include many smaller springs, many of which are not even marked or identified on maps, such as Mullet Springs in the bottom right there. But the really important thing about this water is that since it's coming up from the subterranean aquifer, it maintains just about the same temperature of water year round. That goes between about 21 and 24 degrees centigrade. So that constancy of temperature means that these springs could be thermal refuges for species that are otherwise experiencing temperature shocks. If I'll just give you the example of snook, snook have a lower lethal limit of about 12 degrees centigrade. And we've measured in situ ambient temperatures in the rivers and in the ocean that range from 7 degrees centigrade in the winter, which is well below lower lethal limits for snook, all the way up to 35 degrees centigrade in the summertime, which is considerably hotter than most temperate species can withstand. We know from telemetry work with snook that adult snook are migrating all the way up into springs, such as Manatee, several miles from shore, every winter to spend the winter in that spring in this sort of hot spot of temperature of thermal of water that's thermally warmer than ambient and that is above their lower lethal limit in order to take advantage of that thermal refuge. Manatees use springs in the wintertime as well. Many other species do, and this is well known for Florida springs. But we now have this novel species snook, which is expanding northwards. And juvenile snook settle coastally. They do not make these several mile migrations all the way up rivers. So that led to this sort of open question whether juvenile snook are utilizing coastal microsprings as thermal refuges. And not just snook, but other species may also. So this is some of the flow rates for those springs. Manatee has 150 cubic feet per second, which is one of our largest springs in North Florida, and as I mentioned, are utilized by snook and manatee. But smaller springs, such as Mullet Spring, which is down at 0.25 cubic feet per second, just the same are emitting this warm water, but it's unclear whether or not those micro springs are being utilized by juvenile sport fishes. So our hypothesis is that if we can identify these thermal refuges, then in the winter time, those refuges are going to support range expanding tropical fishes such as snook that would otherwise be evicted from that system. And in contrast, we expect that in the summertime, those same micro plumes of groundwater may support temperate species that are sort of contracting their edge northwards, species that otherwise would be outside of their thermal tolerance window in that 35 degrees centigrade summer heat wave. And so in order to answer these questions, we set out to study springs and control sites around Cedar Key. Here's a zoom in of where those sites are. So each red pushpin is a thermal refuge site, and each blue pushpin is a nearby control, which was paired accordingly to that thermal refuge site in uh, similar hydrology and geomorphology. So here's zooming in on one pair of these sites, two pairs of these sites, sorry. And I'll note that we were actually able to identify candidate thermal refuge sites using a 2005 USGS publication that used thermal drone mapping to identify where these micro plumes might exist along our coastline. So this was done at night in the wintertime when the difference between plume water and ambient seawater or river water would expect it to be maximized. And so anywhere that you see a red polygon, that's indicating that that water is at least 5 degrees centigrade above ambient for that time of year. Blue polygons indicate at least 1 degree centigrade above ambient. And then again, the red push pins are indicating where the thermal refuge sites are, and blue push pins are indicating where those control sites are. To give you some context on our environmental data, this is comparing summer 2025 to winter 2026. And mostly I want to just bring your attention to temperature. You can see that temperatures varied considerably between summer and winter, on average about 26 degrees centigrade during summertime, and on average about 15 degrees in the winter. But temperatures did get down below 10 degrees centigrade, in particular, control 12, sorry, they got down to 7.4 degrees centigrade. So that's really, really t cold and far colder than juvenile snook are expected to be able to survive. Um, just so you can kind of get a sense of what it looks like when we pull up one of these seines. They're, absolutely wriggling with fish. And this was from one of our nursery sites that we identified as being really important for snook. You can see that, I'll just play this video again. Um, Literally like one in every two fish in this seine was a snook. And this was after many, many tries trying to find these little guys and not finding any. So we were just overjoyed by this discovery. And since then, we've found four other important nursery sites. So we're now up to five nursery sites that are harboring really large numbers of juvenile snook. And I think that's really exciting for us to be able to move forward in coming years and monitor how these nursery habitats are used over time. We got a whole bunch of different marine and estuarine species, over 40 species across all of our seines. Here's some of the most common ones. You can see some of our important sport fishes. So that's snook in the top left, spotted and sand sea trout in the middle top, and red drum in the top right. We've also got some kingfish, some gray snapper, and some flounders. So really a good mix of salt, estuarine, and freshwater species, both of fishery importance and forage fishes. Um, Here's the information for our juvenile snook. We got 322 juvenile snook over the last year, which is really fantastic. I'm just overjoyed about that number. It was really, really hard to find these guys. And this is the distribution of their sizes. So you can see that the largest was about 250 millimeters in length. The average was only 88 millimeters, so that's really, really tiny little guys. And the smallest was, I think, 39 millimeters, if I'm not mistaken. So we have a good sort of range of sizes. At a certain point, these guys get large enough that they kind of grow out, they outgrow that nursery habitat and they will migrate out into nearshore waters. And so that's probably somewhere at about 250 millimeters. We also were curious whether size and the relationship between size and length differed between Cedar Key and a farther south, sort of more historic site where snook would normally be found in Crystal River. We didn't really see that. They look like they're kind of growing at the same rate, even though they're in this now colder, temperature constrained location. Another question that we had was um, if we are able to estimate where larvae might end up, then we might have a better shot at finding the nursery habitats that are supporting juvenile snook spawned by those adult spawners that we know about around Cedar Key. So in order to do this, we basically jerry-rigged some very cheap floating passive tracking buoys that are meant to basically do what a larval snook would in the water column, float with the current for several weeks until they find a suitable habitat and they settle out of the water column. So when we put these out in Cedar Key, we really had no idea what to expect. Based on like the loop current, I kind of thought that they would be sent down around the Keys and maybe up along the east coast of Florida. But in fact, this is the track over the course of 3 weeks of one of those larval passive buoys. So what you're seeing here, every red point is a point where that tracker received its location from satellites, but it only has one-way satellite communication, so it receives its in the telemetry of where it is located on the planet, and it stores that information. But then for it to send that information back to us, it has to get within cell service. So every yellow triangle is a point where it got back into cell service and it was able to transmit us that information. So it's a lot to look at, but just what I want you to see is that from the deployment, that initial deployment where that little white arrow is, this tracker buoy, every single zig and zag, so going up and then down, that's a change in tidal height. So it's going in with the tide and then out with the tide. And over the course of about a week, it zigged and zagged its way in and out of the coast all the way down to about Crystal River. You can see the outflow of the Crystal River power plant, Duke Energy's power plant, on the bottom right of the plot there. Then it zigged and zagged its way all the way back to Cedar Key. And then it zigged and zagged its way all the way back to Crystal River. So what this tells us is that this buoy was trapped in a gyre, and that gyre is essentially circulating water between Cedar Key and Crystal River, which is really cool for us because that means that any fish that is spawned off of Cedar Key has a good chance of getting entrained in that gyre. And if we collect juvenile snook from nursery habitats that span Cedar Key to Crystal River, then we have a good chance that that offspring will be from one of those spawners in our region as well. So in the next coming years, we're going to continue doing the sampling and we're going to work with geneticists down at the Florida Fish and Wildlife Research Institute in St. Petersburg to actually do this pairwise close kin mark recapture statistic and estimate the total effective population size of the snook population in Cedar Key. And again, this is something that's not easy to calculate. You need many, many thousands of samples normally for large, widely dispersed marine populations. But this novel, new, expanding species, kind of at the edge of its range, is probably still a pretty small and constrained population. And so, with a reasonable number of samples, we have reason to believe that we will be able to get to that total effective population size. And ultimately, that's the number that we're trying to manage for when we manage a fishery in the United States. We're also doing reproductive biology. So this is working with those adult spawners. So we actually use hook and line surveys at known spawning locations around Cedar Key, and we collect hydrated eggs from the females to confirm that they are spawning. And we also collect fin clips for, again, that genetic study where we're trying to match adults and their offspring. One thing that we found just from this work so far is that in Cedar Key, so snook are protandrous hermaphrodites. So they start off as males, and then at a certain point in their life, they transition into females. That's a normal process. That's what this species does, as many species of fishes do. What we found is that in Cedar Key, they are transitioning into females much later in life at much larger sizes. And so as a result, the Cedar Key population is heavily male skewed. And we kind of have two alternative hypotheses for why that might be. First, it might be a temperature effect, just because they're being exposed to lower temperatures on average than they would normally in their historic range. That might be slowing down that process of sex transformation. Another reason is that it could be a social indicator or social cue that sort of spurs them into transitioning into females, and that they're still at a low enough density in this new population that they just aren't getting to that high population density that would sort of push individuals into becoming females quite yet. So again, we're collecting these gonad biopsies and measuring each fish, taking the fin clips, and then ultimately we'll be able to compare the genetics of the old adult population and the juvenile population. This is what the ovarian tissue looks like. Each of those little cells that has a bunch of points is an ovum, and those will eventually develop into hydrated eggs. We're also going to be working on aging these fish. That's what my student Max is working on right now. Again, we're kind of interested in comparing juvenile snook that grow up in Cedar Key to juvenile snook that grow up farther south in their historic range to understand what are the differences in early life history of this fish in its new range. So we kind of hypothesized that northern snook might grow faster than southern snook. A, because they've got access to potentially more food and fewer competitors, and B, because the growing season is shorter, and so they may have to grow faster in order to get to that size that they're large enough to grow and migrate offshore. So some preliminary findings from our sort of snook focused work. FWC, in collaboration with the Nature Coast Biological Station, has located 9 spawning sites off of Cedar Key. My lab has located 4 young of year nursery sites, so places where, like I showed you that video, we have good densities of juvenile snook that are growing up. Now that's only 4. We visited over 40 sites and only 4 had anywhere more than one snook uh, living in those nursery sites. So that means that they are not that ubiquitous. They're pretty clustered. And so future work will go into trying to understand why those specific nurseries do really well. It might have to do with environmental conditions. It might have to do entirely with currents and where currents are funneling larvae. From those surveys, we've caught and fin clipped 332 young of years. So those are the less than 1 year old snook that are definitely growing up in that nursery. 32 other species that were caught. And again, these nurseries are highly clustered. Some of those nurseries are supporting upwards of hundreds of juvenile snook. And most of the places that we went had 0. So very, very high clustering. We also never found a single snook north of the Swanee River. And now, again, we're just talking about juvenile snook here. Adults are caught north of the Swanee regularly, but we've not found any juveniles north of the Swanee yet. If anybody is listening and they know of potential nursery sites north of the Swanee River, please send me an email. We're really actively looking for them. Currently, our hypothesis is that the Swanee River might actually be a barrier to recruitment. Again, that's where we're trying to get at with putting out that passive buoy and trying to understand where larvae would likely float on their own. And the Swanee River may simply push larvae farther south. These were the most abundant species in these surveys. Sailfin molly was by far and away the most common, but we also got a lot of mosquito fish, a lot of minnows, a lot of Eustenostomus, uh, what's the word? A lot of Eustenostomus species, moharas, a lot of Eustenostomus moharas, and a lot of killifish. So when we look at all these species kind of together in a single plot, this is what is called a constrained ordination. We take each of these abundances of individual species and then we plot them together in a single ordination. So now we're just going to look at 2 axes of variation, and we're trying to compress all of that community variance into just 2 axes of variation. So when you see 2 different points on a plot, each point is representing one entire community, so one of the places where we did our seines. And distance amongst points indicates difference in the community. So the farther apart they are, the more different they are at the community level. And we can also plot on top of that some of those environmental drivers and how they correlate with differences in communities. So here I'm showing you just the results from last summer. So if you recall, we had the hypothesis that in the summertime, uh, thermal refuges would provide a sort of a beneficial habitat to temperate species. And so we would see a predominance of temperate species occupying those thermal refuges. And that in the wintertime, we would see the opposite, and that tropical species would occupy thermal refuges. And if thermal refuges are important at all, or harbor different communities, then we would expect all of the thermal refuge sites to be in one place and all of the non-thermal refuge sites to be different. What we see from this plot, which is again this constrained ordination where we're trying to get information about all the species at once, we see that pattern doesn't really come out. The red points, which are thermal refuge sites, are kind of evenly interspersed amongst the control sites. That means that we're not seeing a strong community difference between thermal and control areas. What we did see was that longitude came out as the most important driver of variation in communities. And why we think that is basically longitude is the left to right axis of this map. And so that essentially correlates with sites that are around the mouth of the Swanee River to the top left of the plot versus sites that are to the south of Cedar Key, to the south, to the bottom right of the plot. So longitude is sort of the variable that best correlates with this river mouth to non-river mouth estuary communities or habitats. And salinity, which you would think would be sort of the biggest driver of that, doesn't actually come out in this model as being that important. Basically, we attribute that to the fact that these are intertidal, and so depending on what tidal height you're sampling at, salinity can change a lot at a particular location. So it just wasn't really that consistent of a driver. But when we actually expand this sampling, so that was just summer of last year. We've now done fall and winter sampling as well. The story starts to get a little bit more complicated. We have very strong seasonal variation. You can see that the now I've plotted summer is the red points, winter are the blue points, and fall are the yellow-orange points. So we can see that fall falls out more or less between summer and winter. So that makes sense that it's sort of this intermediate condition between summer and winter communities. The species that were most different between summer and winter communities, we had in the summertime a lot of Anchoa mitchellii, so anchovies, and a lot of Eusinostomus. Man, this this word is always hard for me today. The Eusinostomus moharas were really common during the summertime, and then Leostomus xanthoris spot was really common in the wintertime. In the fall, we actually had a lot of Poecilia and Fungulus and Cyprinodon that came up in our sampling. But right away, we're seeing that these refuges are being occupied by very different species depending on time of year. When we zoom in on just the sport fishes, these are just six of the dominant sport fishes that we saw as juveniles. So Centropomus and decimalis in the top left, that's snook. The top two right plots are Cyanocyan arenarius, that's the spotted sand trout, and Cyanocyan nebulosus, the spotted sea trout, on the top right. Bottom left, Metaceres species, those are kingfish. Bottom middle, Mugelcephalus is mullet, and bottom right, Cyanops ocellatus is red drum. So right off the bat, you can see that there's sort of three different syndromes or distributional patterns here. The two species of sea trout and the kingfish were all more abundant during summer months. So that reflects their summertime utilization of nursery habitats. So they're spawning in the early spring and summer. Those juveniles are growing up and then leaving the estuary by fall. In contrast, snook are common in the fall and into winter, and then mullet and red drum we really only saw in the winter. So just based off of this information, we have evidence that snook could be competing with red drum and mullet for space utilization. Mullet have a very different food apparatus and food that they seek out. They're planktivorous. But red drum feed on a lot of the same things that snook do, and so there is the potential for dietary and certainly spatial overlap of these two potential competitors at this early stage of their lives. When we look at temperature results, there was actually some equivocal temperature data, and I'm going to get into that in a little bit, but this site showed the best and clearest pattern that was basically what we hypothesized would happen. If you look at the bottom plot, that's just showing the temperature difference, and anything in blue indicates that the control sites were warmer than the refuge sites, and in red, that means that the refuge sites were warmer than controls. So as we start at the beginning of the plot, that's September, we're coming out of summer. At that point, controls were warmer than refuge. So again, the refuge site is a thermal anomaly. It was as much as 5 degrees cooler than those control sites. And as we move into winter, that trend flips, and we suddenly see that those thermal refuges are warmer than control sites. And these were almost up to 10 degrees centigrade warmer than control sites. So this is evidence that thermal refuges, these micro springs along the coastline, are presenting a different habitat than ambient, and that the difference itself is switching based on season. So again, I think that this is environmental evidence that these thermal refuges could be providing a very different habitat requirement to temperate versus tropical species depending on season. Some of the major takeaways, we've seen that fish community variation and environmental variation was driven by season and primarily, but also by some environmental and spatial drivers rather than necessarily a thermal refuge versus control comparison. Although there was one site, this S11 versus C11, where we did see a really strong difference in line with what we hypothesized. But in general, those thermal refuge sites did not differ significantly in temperature relative to control. We also didn't see any snook occupying thermal refuge sites during winter months. None of those four nursery habitats that we identified exhibited that thermal refuge characteristic. So it's not necessarily just driven by that. But we did see that juvenile native sportfish species, in particular the cyanocyan sea trouts, occupied those thermal refuges during summer months. But this is likely due to their natural seasonal spawning patterns, more than necessarily seeking out those particular thermal refuges themselves. So why wasn't there this temperature variation? Well, we're actually in the midst of a 25-year historic drought. You guys have all seen the burn bans and the wildfires that we've been experiencing. So when the swamp is on fire, that's a good indication that we're in a pretty bad drought. And drought means a lack of aquifer. And it could be that these micro springs are, low flow normally, and if the aquifer height is low enough, then they might not be reaching that sort of critical threshold of height at which water would start to emanate into these coastal habitats. So in that context, it's good to see sort of the historical pattern of rainfall in Levy County. You can note that the thermal imagery that was taken by USGS took place in the midst of a wet period that immediately followed another wet period. The data that we've been collecting now are in the midst of a very, very long drought that was preceded by another intermediate sized drought. So that tells us that these microplumes as thermal refuges are probably ephemeral. They are probably dependent on aquifer height. And this gives us a little bit more information and perhaps ammunition to defend the rights of fresh water. The amount of fresh water that we're taking out of the aquifer ultimately will impact the long-term sustainability and success of these fisheries that we really care about. And if we pair up that drought data with the pattern of snook expansion around Cedar Key, which is from Caleb Pertobal et al.'s study in 2020, we see that there, if you squint your eye at it, there could be a pattern here where we see this sort of uptick initially in snook numbers that corresponded with a wet period in the late 2000s, and then another big wet period in the early 2010s that corresponded with the exponential increase of snook in our region. So this is, correlative evidence at best, but certainly from the side of the juveniles that are growing up in nursery habitats, the amount of fresh water coming into coastal microsprings might have a really important influence on their success and survival and recruitment into adult spawning populations. So as we go forward, we're going to be again doing close skin mark recapture analysis of the snook population around Cedar Key to determine the structure of the stock and to analyze how many parent offspring pairs we can find. And using that, we can estimate the total effective population size of Cedar Key snook. We're going to continue with our community ecology of fishes within these juvenile nursery habitats and try to span these dry to wet periods and really get a sense of how ephemeral these thermal refuges are and whether they're more or less important depending on if it's a dry or wet year. As I mentioned, my student Max is working on right now. He's working on aging snook from Cedar Key and other southern estuaries in their more historic range to compare things like age, health parameters, diseases, and parasite loads. And I also just want to say one thing, which is that a couple weeks ago, I was working offshore down around Sanibel and Marco Island. And coming in from the ocean, you could just see this wall of white. And it's essentially just all built up, right? And coming in from Cedar Key offshore, you see a wall of green. And that difference was just so stark to me And I think that it's something that we should continue to think about as we're trying to uh, develop the nature coast in a responsible and sustainable way. Let's keep that coastline green, in particular for nursery habitats for these important sport fishes and other species that really matter a lot to us. So with that, I'd like to thank you all for being here. Thank the folks in my lab and my collaborators at FWC and the Nature Coast Biological Station. And one last time, I'd like to thank Laura, Savannah, and Fia for organizing this talk series, which I think is just so valuable. So thank you all.
Awesome. Thanks so much, Robbie. That was really comprehensive. So cool to see everything that's going on. Um, So, folks, please pop in the chat if you have questions, or feel free to raise your hand if you'd like to come off mute. I do have one that I can start us off with because sometimes people do the poll and then they put their questions. So, um, a while ago, I want to say it was almost 10 years ago now, there was a master's student named Emma who worked with several of the fishing guides along the Nature Coast to collect fin clips from adult snook. And I was wondering if that project has sort of come up in the context of the fin clip work you're doing, and if there's any sort of utility for that from that old genetic survey to kind of inform the new genetic survey in terms of effective population size. Like if you would be able to look in and see if there's population growth or um, anyway, just about that project.
Yeah, I wasn't aware of that data set, but I'll look into it. It's likely that my collaborators at FWRI were involved in that fin clip collection. Right now, is the fact that we now have access to the genes. You really need both the adults and their offspring, and enough pairs of parents and their little offspring. And so, with enough pairs relative to the total number sampled, you can estimate that reciprocal gene flow and estimate the total effective population size. So that's something that we could continue to monitor over time into the future. Not likely something that we could backtrack to 2010 on, but it will give us information on like the survival of the spawning adults. We don't, quite yet know how long they might live and how many offspring they might reproduce over the course of a lifetime. So that certainly would be helpful to understand sort of the long-term survival of those individuals. Also, there's sort of this founder effect where some individuals of the southerly snook population migrated to Cedar Key and established there. And that is a subset of the genetic diversity in the parent population. So that would be another cool comparison to make, whether the sort of allelic richness, the genetic diversity of modern day adult snook and Cedar Key has changed relative to that 2010 collection. So I'd love to get the contact of that person when you get a chance, Savannah.
Yeah, absolutely. I saw Fia's hand go up, and then we have another one after.
Hi, yeah, great talk, Robbie. I was wondering, since you talked about how like maybe some of these areas are acting more ephemeral, um, when you found your four sites that like consistently had the juvenile snook, um, was that like just based on 1 year of sampling or like you were consistently finding them?
So two of those locations we sampled both this winter and the previous winter. So those two at least I can say are consistent. The other two we found for the first time this winter. So I don't know from year to year how much those might change. I will note that the best nursery habitats that we've found so far have not been in places that we would have expected. They've been right up next to roads. They've been at like culvert sites, places where there's like trash in the water that people throw off the side of the highway. Um, so I think that actually speaks to the fact that places that we don't necessarily think are valuable might be really valuable. And on the one hand, we should clean them up, and on the other hand, we need to identify them so that we can put protections into place before development really starts to ramp up in the Nature Coast area.
Cool, thank you.
And Mark also has his hand up.
Hey, Robbie, great work, man. Great to hear all this stuff get put together. Especially the gyre stuff. I want to talk to you about that later. But I'm curious. how frequently does like the tropicalization of a predator result in a shift in ecological role or like trophic position? And are we thinking that we're going to see that, or that we're seeing that with snook?
Well, certainly there could be shifts in the trophic position or what the diet would be of this invader, because they're now in a place that they didn't exist before. You would expect that something about what they eat will change. I think a more poignant question for our region is, um, are they eating things that other fish would like to eat? And so by doing so is the introduction. It's not an invasion because this is a natural expansion of a species that already existed in Florida's coastline. Just that we've kind of changed the environmental conditions to allow it to expand its range. But I think the more poignant question there is it eating things that other native species to the northern Gulf would also like to eat? And if that's the case, maybe there's the potential for competition for those limiting resources. Um, I know that folks are doing this with adult snook right now, but we don't really know anything about the juveniles, which could have kind of a fundamentally different dietary overlap with the juvenile sport fishes that are native to the northern Gulf. But in terms of range expansions or invasions of predators, the lionfish expansion and invasion of the Caribbean and the Gulf are a really good example of that. They can decrease recruiting reef fishes. Again, these really small guys that are just settling to the reef by upwards of 80%. So that's obviously bad for the populations of those reef fishes that are trying to settle, but it's also challenging for other predatory species that might also want to eat those juvenile fishes as a food resource. So that's exploitative competition where they're trying to get after the same food resource. But there's also like the utilization of potential utilization of thermal refuges. So there might just be like limiting space in a pond or in one of these plumes. And so you can also have this sort of interference competition where simply the physical presence of these new snook in that space prevents the physical presence of some other species that might want to utilize that space. So I think all in all, the snook expansion in the Nature Coast area is probably a net positive because it creates a lot of opportunities for fishing and so on. But we have to be cognizant of the negative impacts too, and that's kind of what we're trying to get at here.
Awesome. Well, I'm not seeing any more hands raised or any more questions in the chat, and we are pretty close to our time. So I just want to say thank you again so much, Robbie, for coming and presenting this, and I hope we can have you again back on Nature Coast Currents. And for everyone else here, we do have one more before we take a break for summer field season, and that will be kind of earlier in the month than usual. It's going to be on June 3rd, which is a Wednesday, and it'll be Amanda Chappell, and she's going to take us to Tampa Bay and talk about some of the eutrophication impacts there. And I believe she's also going to cover the Piney Point disaster, so that should be of a lot of interest to many of our folks. So thanks again, Robbie, and we'll see everybody next time, hopefully.
Thank you. And again, if anybody knows of other juvenile snook hotspots, just send me an email.